And it appears that we're officially live. Hello, everybody, and welcome to another session of our Sussex Vision Seminar Series, as always within the Worldwide Neuro Initiative. I'm George Kafetsis, a master's graduate from Thomas Euler's group, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from University of Chicago, Professor Stephanie Palmer. Following her chemical physics undergrad from Michigan State University, Stephanie joined John Chalker at the University of Oxford for her PhD in theoretical physics entitled Order and Disorder in Two Geometrically Frustrated Antiferromagnets. And it was during her postdoctoral years that an interest in neuroscience started manifesting. First, together with Kenneth Miller in UC San Francisco, and then in Princeton with Bill Bialek and Michael Berry as her advisors. In 2012, she started her group at University of Chicago and has remained there ever since, nowadays as an associate professor at the Department of Organismal Biology and Anatomy and as a visiting professor in Princeton. Stephanie's interests center on population codes, from encoding the input to performing different computations. Throughout the years and in close collaboration with many experimentalists, her work in a variety of systems has provided a number of different key insights, from neurons being optimized for predictive coding to combinatorial or highly dynamic and behaviorally dependent codes, of course, depending on the systems of study. Personally, I'm very excited to be having Stephanie here with us today, presenting some of their latest and I'm sure exciting work in her talk entitled How What You Do Shapes What You See. So without any further ado from my side, please all welcome Professor Palmer. Stephanie, the stage is officially all yours.
Wonderful. Thank you so much, George, for that kind invitation. And it's a pleasure to be here. And I want to give another shout out to Worldwide Neuro. I think this is a wonderful platform and a wonderful way to bring us all together and to continue it so that we have more access across the globe to each other's work. Today is Memorial Day in the US, where we reflect on the horrors of war and the sacrifices that people make. And I wanted to take a moment to reflect on the wars that are ongoing in the world and to think of the people who are affected by that and the people who are suffering incredible loss. So I wanted to take a beat to say that. And also to thank some of my own family members for their service. This is my grandmother, Mary Jane Palmer. I usually talk about her and the fact that she was a physicist at the University of Chicago, where she got her undergrad degree back in 1941. She was an inspiration to me and also makes me feel good about the University of Chicago because they admitted women back then, which was unusual. But I also wanted to share today that after she got her BS in physics, she worked on radar during World War II at a naval base in New Jersey and was one of those brief period where women were accepted for volunteer emergency service in the US military and did what she could in her part as a physicist to help with that war effort. So just again, thinking of the people who are being affected by war right now. Okay, so taking a pause, that was a little heavy, I know. But important. What I'm talking to you about today is how what you do shapes what you see. I'm highlighting a few of the group members here who provided key data, calculations, theory, and insights into the story I'm going to tell you, and highlight two postdocs, Jared and Sue, who are moving on to new things now. Jared doing a second postdoc, and Sue is a senior postdoc in the group, and I want you to keep an eye on this space for those two names. I think you're going to be hearing wonderful things from them in their work going forward. Okay, so I want to challenge you to think about whether or not any biological system is actually optimal, because we as biologists know that evolution is not the theory of optimal. Evolution is the theory of good enough. Natural selection lets you do better in a fitness goal, but doesn't necessarily try to drive you towards what a theoretical physicist like me would think of as optimal. So why have we had so much success finding optimal behavior on neural systems? I would argue that it's because those who came before us, people like Simon Laughlin and Horace Barlow, knew and intuited and thought very carefully about this question: what matters to the biological processor? So when you want to think about optimization in the brain, you have to think not about what you would do if you were that sensor, but what the behavioral goals of the organism needs to accomplish its sort of mission critical tasks. What I want to take you through today are some of those mission critical tasks and how we've been able to dig out a little piece of this puzzle about where and when neural systems, particularly in the retina, are optimal. So the way that we think about this is within the framework of optimal coding. We think about maximizing information transmission subject to constraints. So this seemed a little bit mathematical and maybe a little abstract, but what I want to unpack for you is that those constraints right there are embodying the behavioral goals of the organism when this kind of optimal coding picture is done really, really well. And so taking inspiration from early theoretical neuroscientists, we're going to try to tune this up a little bit and take the next steps. So weaving those behavioral goals all the way back into early sensation. So one of the behavioral goals that I'm going to pitch to you today pretty hard, it's our bread and butter, is prediction. We like to think about how the brain overcomes some of the crumminess of its parts. So the fact that neurons have noise, so we could argue about whether or not that's a bug or a feature, but all neurons also have delays. And delays are kind of hard to wrap your head around as a feature because those delays operate on time scales that would be really hampering for biological function. So the classic example of this is to show a professional sports player and talk about how well they do anticipation. So my favorite sports player is Serena Williams. This is Serena back in the day at the Rogers Cup. And this is illustrating that she's trying to hit that tennis ball. And if she didn't compensate for the lags in her retina, which are even as short as 50 milliseconds, which doesn't sound like much, but if that ball is traveling, okay, let's use sensible units. We're in the UK right now, nominally. So if it's traveling at 100 kilometers per hour and there's a 50 millisecond lag, that's more than a meter of difference between where the ball is and where a laggy retina output would say it would be. But of course, when you look at this, you see that Serena is looking right at the ball, not back over here. And if you look closely and track Serena's eyes, you'd see she's actually looking at where the ball will be. And this isn't a superhuman feat. I mean, we could argue that Serena is, a Six Sigma event in human history in terms of being able to play tennis, but you and I also do this. And we could certainly see where the ball is going. That's not a very fast tennis ball at 100 kilometers per hour. We just couldn't get our racket on it necessarily. And that's where Serena really shines. So what I want to argue to you today is that this kind of prediction for predictable motion of objects in the visual scene starts as early as the retina. And I know this crowd there in Sussex, strong believers in the awesome power of the retina, of course, because over the years from seminal work back in the day all the way through current research, we know that the retina is an incredible computational device. It's a piece of your central brain and your peripheral nervous system sitting together in the eye to do a lot of pre-processing for you, even fancier than what our phone cameras do. So if you take a slice through the retina, there's this beautiful and general circuitry across vertebrates with a photoreceptor layer, the guts of the retina, and the output ganglion cells. Today, we're going to be focusing on these ganglion cells and trying to understand what happens to the light signal from soup to nuts, so from the photoreceptor layer down through this output layer. And some beautiful work from Thomas Euler's group shows us that there's just a massive diversity in functional types in the ganglion cell layer. And no matter whether you're a lumper or a splitter, you can't avoid the fact that there's lots of different fundamental computations going on amongst the cells in the output layer. So biology doesn't build stuff for nothing, and it certainly doesn't build stuff to suit our expectations, but it doesn't, have any drive to, carry around a bunch of vestigial circuitry that's this complicated without a purpose. So we also know that lots of really important, a rich set of complex computations happen in the retina, and things that are related to prediction. So when Michael Berry was a postdoc with Marcus Meister, he looked at motion anticipation in the retina. And this is the fact that when an object is moving in a steady and stable trajectory across the eye, the neural image of that motion actually is pushed towards the leading edge. So this is for, say, a bar that's moving to the right here. The red is where a flashed bar would appear as a neural image on the retina, and the blue is where the neural image appears when it's moving. So you see it's sort of skewed. And there are many other nonlinear sort of motion effects like this. The reversal response, which Greg Schwartz worked on when he was a graduate student with Michael Berry, where a whole population of retinal ganglion cells fire simultaneously when an unexpected reversal happens in an otherwise smoothly moving object. There's the omitted stimulus response. There's object motion sensitivity in the retina. And there's even, with this motion anticipation story, lag normalization that Gautam Aratramani's group showed beautifully happens because of gap junctions between direction-selective ganglion cells. And all of this is happening in your humble eyeball. So what we would like to do is ask if this general problem of prediction that all of these individual results hint at can be quantified. So I'm going to take you through a little background on how we approach the problems. I'll keep the equations a little light, but point you to some papers if you'd like to know more. And then we'll dig in to some more recent results on how we're trying to peel apart how behavioral goals relating to prediction sculpt the visual circuitry in the retina. So we use the information bottleneck method to quantify how much a retinal ganglion cell code knows about the future. So here's an illustration, a cartoon really, of this bottleneck. You have some stimulus information coming in. That gets encoded in the ganglion cell firing here, which we're calling the word, the neural word, ones and zeros, so spikes or not spikes, from a collection of ganglion cells at the present. And typically we'd say, how does the past lead to these spikes? But here we want to say, how does it also give the brain information about what's going to happen next in the stimulus? So we are trying to compress our representation of the past because the brain has limited amounts of ATP and limited number of neurons. So there's compression, but it's not compression just to do some sort of as close to lossless as you can. There's a willingness to throw something away. And our hypothesis is what you throw away is the stuff that's not predictable, so that you retain as much as you can about the future. That's a nice thing to say in words, but you need to be able to put an equation to it so you can test quantitatively whether or not the real retina does this. So we can quantify this using this Lagrangian, and it's a trade-off between two terms. So if we want to minimize this, we're going to make this first term small, and this is the compression part. This is the mutual information, or just a generalized notion of correlation between what the brain is saying now and what the stimulus was in the past. And then there's a minus sign here. So this is the part we're trying to hang on to. This is the part we're trying to maximize. We want to keep beta amount about the future, where this is what the brain's saying now, and this is what the stimulus is doing in the future. So that right there embodies our notion of the behavioral constraint. In the bottleneck world, we get to put anything we want in the second term, and we've said what matters is the future. So we're going to throw stuff about the past away, but we're going to try to keep as much as we can for prediction. So typically in a neuroscience experiment, if you want to study motion in the retina, you might do this. You might show the retina a steadily moving bar, or you might get fancy and show the retina something that reverses course. That's all well and good. One is completely predictable and one is completely unpredictable. What we'd like to do, though, is give the retina a choice. We want to say, here's predictable and not predictable. Show us what you throw away. And our hope, our expectation, is that when the retina shrinks down its representation, it preferentially throws away this unpredictable part. So again, we have to quantify that. And the first thing we need is a stimulus that meshes together this smooth, predictable motion with unpredictable bits. And I keep saying bits, and I'm trying not to be punny, but there you go. All right, so here is the movie that we showed the retina back in the day that led to our first set of solid results. This is a bar that you can see is kind of jittering around, but has like a kind of predictable component. You know it's not going to get too far away from the center of the screen, and it does make some excursions where it moves for a little while in the same direction before it gets kicked. And you can kind of intuit that the kicks it's getting are noisy. It doesn't seem, maybe it even seems to you a little bit like Brownian motion, and you're recalling all of your intro physics classes because it really is Brownian motion. So this is dv/dt, or the acceleration of the bar. And just like F equals ma, we've got a minus v over here. This is like a drag term. We've got a fictive spring characterized by omega naught that tethers the bar to the center of the screen. So x is the position of the bar, v is the velocity of the bar. And here's that noise term. Gamma of t is just a white noise process, and d is kind of a diffusion constant. So all of this governs the motion of the bar, and parts of this are predictable. So if you're just looking at the green parts of this equation, you're solving a damped harmonic oscillator. And now everybody loved physics 101, so you remember how to solve this exactly, right? But then this blue thing is the unpredictable part. And this gives it kind of like a Brownian motion-like feel, but with this restoring force that keeps the motion at the center of the screen. That's important for us because we're going to show this exact movie to a salamander retina. And we want the motion to stay around the cells that are at the center of the array. This is an image courtesy of Ronan Segev when he was a postdoc in the Berry lab. So these are backfilled retinal ganglion cells, and these are the electrodes. We can make much bigger arrays now. This is an image that was from data we collected with the Olivier Mars group in Paris. And here you see a nice chunky array with this 30 micron spacing. And we sent a student over to Olivier's group to record from the rat. So that's what we're going to do. And we're going to show that kind of bar movie. And here's what happens. So first of all, here's a sketch of the bar motion up and down on the screen. So this is just the center position of that bar. Here are some spikes from some cells. Here's the time at which those spikes occur. And as a function of delta t relative to where the spikes occur, we ask, how much does this retinal word know about the position of the bar? So way back in the past, you don't know much about where the bar is, and in the future, you don't know much either. But as you get closer and closer to where that cell spiked, you get more and more information. And so if we average over many cells from this salamander retina, we see that we get a peak in information that's a little bit lagged relative to where the spike was. So we see back into the past a little bit more clearly than we see into the future. But you see there is this information that bleeds out into the future. And that's what we'd like to interrogate. So this is for single cells. We can do it for 2 cells, 3, all the way up to 7. The reason why this is going down is because I've normalized it by the number of spikes. So if you have more cells, you have more spikes. And what you see here is that those more spikes are actually giving you redundant information about where the bar was in the past. But this rainbow of curves actually flips over and maybe even becomes synergistic out here in the future. And what we'd like to know is whether or not this bleed through into the future is as much as the retina could get, given that it knew this much about the past. So again, we're going to come back to our bottleneck cartoon. We've said that what the brain wants to do is do this predictive computation. But of course, that's going to be constrained by the stimulus that we put in. And here's where the finesse of having, a little bit of background in mathy physics comes from. We know that we can actually solve this equation because this motion is simple enough. So this equation becomes solvable. Awesome, very happy about that. So we can say, if the retina were optimized, what would its trade-off look like? And that looks like this. So here what we've got is, again, we're showing this bar movie to the retina. because there are correlations in that bar, we can calculate how much you could possibly know about the future, given that you know some amount of the past. And that's what this bound curve is. So the gray part is the forbidden region. The brain can't get there, but this is the optimum. So as you compress more and more, you're moving this way along the x-axis. And if you have no information about the past, you can't know anything about the future. That makes sense. And you can never know more about the future than you do about the past because, you know, no fortune telling and all that stuff. We're standing on a lot of previous work, particularly from the late Tali Tishby, Bill Bialik, and Gal Chechik's fantastic paper on how to solve exactly these kinds of problems where it's a Gaussian information bottleneck problem and you can solve things analytically. And if you'd like to know more, here are some later papers from our group that dig really into how to solve these equations. But we're here to talk about the retina. So the retina gives us access in the firing rates and the length of the experiments we can do to this part of the bound curve. And if we zoom in there and we plot, again, the bound, that's the optimal trade-off, but then we want to put the retina on this plot. So we plot how much the retina knows about the past and how much the retina knows about the future. And in the salamander retina, for 1, 2, 3, 4, 5, 6, or 7 cell groups, we get really, really close to the bound. And that was like a preliminary result that got us going on this bottleneck approach to thinking about prediction in the salamander retina. It turns out that when Jared Salisbury went to Paris to work with Olivier, he also found that this works in the rat. So in the rat retina, he plotted the information about the past, information about the future, and showed the rat retina a similar kind of movie and found that this bound was again saturated. But there's something really clever that Jared was able to do in the rat retina. And because he was sitting in the MAR lab and had, you know, experts around to help him get a really good recording, he could play a couple of movies to the same rat retina. So he could explore the space of motion. So the first was something like what we showed the salamander. You'll notice that the contrast is flipped. The salamander retina is composed mostly of off cells. Here we've got a rat retina, so we changed the bar from a dark bar to a white bar. And he also showed this other stimulus. Now, what's interesting is that I'm sure you can see that this seems to be getting, you know, sort of, it's getting kicked, but it seems to have more mass, seems to be oscillating maybe a bit more just by eye. I promise you that both of these movies explore the same spatial profile and same velocity profile. So if you were just looking at snapshots in time without the whole movie, you would think that you could not distinguish between these two movies. The point here is that motion is really, really the essential element in the retina's response to these two. And the way that these two differ is in their ratio of these predictable components to their non-predictable components. Actually, it's just in the predictable components here. So this stimulus is critically damped, and this stimulus is underdamped. And what that lets us do is explore whether or not the rat retina is solving all of these prediction problems equally, or if it has particular prediction problems that it's evolved to solve. So what matters to the rat is the question we're asking here. And what we find is that for stimulus 1, which was like the one we showed the salamander, the neurons are definitely not at the bound. And for stimulus 2, which was an underdamped motion where velocities are a really important key element in prediction, we're really close to the bound. And this was fun. And we were really happy that we didn't just keep seeing the retina smashed up against the bound every time. Because if we did, we'd start to worry that we were fooling ourselves, that either it was too simple a prediction problem or that our calculation was just sort of messing us up somehow, that we weren't looking in the right place. We actually want to see our notion of optimality fail sometimes. We want to be able to prove ourselves wrong. And so we were glad to see this. The other reason we're glad to see it is that this retina still cares about this motion because it has lots of information about the past. It just doesn't predict it that well. So what we think we're seeing is a tuning of the filter properties in the rat retina for this kind of motion. And I could wave my hands and give you a just so story about why that is, but I want to move on and take you through some more research. What Jared and in collaboration with Olivier and Kyle Boyanek in the group have been able to do now is to show 5 different movies back to the salamander retina. And we're trying to tease apart exactly what components of motion get embodied in the retinal prediction best and how that mechanistically is underpinned by the circuitry in the retina. It's important to think about the circuitry in the retina because we want to know whether or not this kind of behavior where you're close to the bound is easy to achieve. So we have shown that a classic linear nonlinear Poisson model, our like textbook picture of the retinal ganglion cell, does not saturate the bound. So the real retina does a heck of a lot better. And we tried to give this LNP model every advantage we could. The only thing we didn't do is let it have correlations between neurons, and we didn't let it have like adaptive gain. So we think that we could add some single cell properties and maybe some collective properties in to move from the LN model to the data. And Kyle in the group is working on that. And he's awesome, and you should watch that space. Also, keep an eye out for some new Salisbury papers, probably hitting bioRxiv this summer. Okay. Good. So we got a baseline layer. We've got like a little like lasagna results coming into here, and we've got like base noodles. And now we want to elaborate on it and get a little bit more fancy. So moving through the natural world is complicated. It's not like you just you know, kind of objects move in front of your eye and you get some time to anticipate where they're going to be. In a sense, playing tennis, you know, just to hit the ball, not to hit it in some spectacular way, but just hitting a ball is a pretty simple thing. And when we're all moving through the world, more complicated stuff happens. So say that there is some like terrible, scary, terrifying beast that's trying to chase you, and it just leaps out at you from behind a barrier. That's something that you would experience in the natural world potentially. I hope not. I hope everybody, you know, sees no bears today. But imagine you saw this creature just run at you from behind a barrier and then run behind a barrier and then come back around. Your visual system is actually really good at predicting where that terrifying object will be. This is our puppy. And what we wanted to do was to think about how this works in the retina. This is a collaboration with Wei Wei, who is a fantastic retinal researcher, runs just a fantastic group here at the University of Chicago. And what Jen Ding did when she was a grad student in Wei's group was explore classic bar motion, but then she put a little riff on it. Jen had things go behind a barrier. So this is a pretty simple riff on the typical directional motion selectivity story in the retina. But it embodies something about that complex video I showed you of the puppy running around, where he ran behind barriers and behind a wall. And you could anticipate where he was going to be when he came out. And it also probably surprised you at the very beginning when, you know, this cute little fluffy puppy came out from behind the barrier. So what we want to understand is how real retinas handle that kind of surprising emergence. So what Jen did in, again, Wei's group was to look in the mouse retina. So that's another species here. So we're looking in the mouse retina, full field motion of the bar, and you get the typical direction selective response. So if the bar is moving to the left, you get in the preferred direction, you get a lot of spikes. And if it's moving to the right, you get very few spikes, if any. And then you see the typical DS-ness on this polar plot. What happens when the bar emerges from behind an occluder is pretty crazy. So if you're on the preferred side here and you have motion, so you look at what the response is when the bar is moving to the right or to the left on this side of the cell's receptive field, when it comes from behind an occluder, you find that you get the preferred direction motion, but you also get almost as much spiking in what used to be the null direction. So what happens is that these direction selective ganglion cells actually just totally lose their minds. When something emerges from behind an occluder, they lose their DS-ness completely. And what Jen did in this paper was to take this apart by doing an incredible series of experiments to pin down the dendritic arbor of these DS cells, to find out. She did spot stimuli to figure out what their excitatory and inhibitory receptive fields looked like. She did EM with the Burson group. She really dug into this at an exceptional level. This was a tour de force in different ways to dissect this effect. But at the end of the day, what Wei wanted to understand was, well, what does this mean downstream if your DS ganglion cells are losing their mind and now suddenly just responding to every direction of motion, in particular their null direction of motion that they usually fire almost no spikes to? What would that mean for the downstream circuit? So one piece of anatomical data you need for this is that these particular DS cells in the mouse retina project to the superficial layers of the mouth superior colliculus. So that's important. So they can trigger rapid movements to orient and things like that. So what we asked was, what would happen if you had the bar moving across a population of those DS cells we saw before, and ones that preferred the opposite direction of motion. So let's say the red and black cells prefer opposite directions of motion normally. But then when the occluder comes in, so these cells, just to say, would prefer left and right motion normally. So they have no business firing together. They usually would never ever fire together because they prefer, things going in opposite directions. So what happens when they do fire together? Well, that's because there's an occluder, and so something that's coming from behind the occluder makes the one that prefers the leftward motion all of a sudden fire a bunch. And the ones that prefer the rightward motion, they fire as they should because the bar is moving in that direction. And that concomitant response screws up, of course, your direction decoding. So here's how much your root mean squared error is in degrees of direction of motion of the bar as a function of time since you emerged from behind the occluder. So here's the time where the bar is behind the occluder. If it's full field and there's no occluder, then you just have this nice low 10-degree discrimination of direction of motion from the population. If you go behind the occluder and come out, you, for this brief period, completely lose the plot. All of a sudden, you cannot tell where that bar was. And that's because the ones that are supposed to be so good at direction selectivity have lost their mind and are responding to the wrong direction. So if you're a static decoder, you don't know that. You don't have an independent measure of the visual system. So your root mean squared error and unraveling the direction falls apart. But there's something cool that happens. When these two cells that prefer opposite directions of motion fire together, it means that there was motion in the overlap between their receptive fields. So that synchronous firing only happens when the motion is in a place in their receptive field that's overlapping, and the part of the receptive field that the ones that are saying the wrong thing is a much smaller portion of the receptive field than you would get for full field motion. So what you get is a really, really fine high definition position signal. So here's the root mean squared error in position in visual angle. So you have with full field motion, you have about a 3.5 degree resolution in where that bar is. After something comes out from behind the occluder, you actually get a super, super duper sharpening of where you think that bar is located. And the reason is that the synchronous firing between the two populations hones down where that bar could be. And so we think that what's happening here is that when an object comes out from behind an occluder, you might not need to know as much about where it is, about what directions it's going, as you need to know where it is. Sorry, you really want to know where that new thing is because it could eat you. You're a mouse, lots of stuff eats you. So what we think might be happening is that these cells are sending their signal to the superficial layers of the colliculus so that you can take this kind of high definition spatial cue and reorient your attention, your body, your eyes towards it so that you can decide, is that a threat or not, and then make your escape if you need to. Okay, I'm going to take you through one more example about how prediction sculpts your optimal processing in your eye, but we're going to move away from vertebrates now, and I'm going to tell you a little bit about flies. This is something I have talked about before in this series, so I won't belabor it too much. But what we're trying to do here is to really take on complex natural behaviors. And since the last time I showed this to the worldwide neuro audience, we have spiced up the figures and have some new pieces of results. So let me give you the update. But here's the world we're living in. We're thinking about ourselves as a little Drosophila, some fly who's moving through the natural world. And this movie is slowed down 40 times. So imagine how fast this little thing is going. And then all of a sudden there's a threat that appears. And remember that Drosophila get eaten by dragonflies. So what this fly does is a banked and counterbanked turn. You can see all kinds of wing movements, the body moves along the way, head movements, and it orients itself directly away from the visual threat. These are beautiful, publicly available data from Michael Dickinson's group. One of the awesome things about our modern era of neuroscience is that when you publish a paper, you have to publish your data. And that gives others an opportunity to use it and do new things. And so Sui Wang in our group was able to take these escape flight trajectories. We can watch it again if you like. From many, many different flies, many different escape trajectories, and use them to make a model of how this is happening. So what we wanted to ask is whether or not the fly's visual system helps it do this fast escape behavior by making a prediction. So let me show you a little bit about fly feedback and escape behavior. So here's that whole banked and counterbanked turn that the fly uses to reorient. That's the evasive flight maneuver. That happens in 40 milliseconds. It's crazy, crazy, crazy fast. Our hypothesis is that the visual threat leads to a feed-forward visual prediction that helps guide this ego rotation in this gap here of 20 milliseconds before you have time to put your haltier feedback, your little gyroscopes, the second wings on the fly that act as gyroscopes and steer, and well before you can have visual feedback, like I'm going to move and then I need to see what happened when I moved, that there is a visual threat that leads to visual planning that gives you a prediction that essentially lets you guide and control in this moment. And the reason that's so key is that flight is aerodynamically unstable. And if the fly doesn't know to turn up the gain on its halteres, it won't do the compensation right when it does this incredibly fast turn. So these turns are also 10 times faster than what the fly usually does when it's just moving really fast already throughout the world. So flies already move really fast and make turns. When it needs to escape, it dials that up by a factor of 10, but it takes time for that feedback to go through. So again, the idea is that visual prediction puts this in place. So working with Axel Borst and Idan Segev, Siwei was able to do all of this in silico. So she took those natural fly trajectories, those natural escape behaviors from the Dickinson group. She took everything we know from Axel and Idan, who are world's experts in the fly visual system, especially in the vertical sensing system. So how you sense up and down, which is what you have to do to make these turns. She played those escape behaviors, those really complex trajectories, through a fly that's flying through a natural world. So this is all painted with Van Hatteren style natural scenes. So the structure of these trees and bushes is like the structure of natural scenes. So there are correlations that the fly needs to disentangle so they can figure out where it's heading as it makes this escape. So we also used data from Axel and Idan to look at the circuitry, the dendritic and axonal circuitry in the fly vertical sensing system. So as it's making these banked and counterbanked turns, how the world is moving up and down, usually we think about the horizontal sensing system in flies, this is the vertical, matter a lot. In the fly, there are 10 VS cells that form their vertical sensing system that survey different parts of the visual world. The central 3, it was found by Dickerson, actually projects down to the neck motor center and could really influence flight. So what Siwei did is she modeled this whole visual system. She put the natural inputs through, the natural behavior against a background of natural scenes as the input to this whole circuit. Because of awesome work that came before us, we know what all the values are and all these resistors and capacitors and the whole bits and bobs of the circuit. And then Siwei could tear the circuit apart and see what mattered. First thing she did was quantify prediction. So this is, again, the information the fly vertical sensing system has about the past and the information it has about the future. This bound you can't calculate analytically anymore. So we needed a computer scientist, that's Siwei, to tackle this and be able to solve for this bound. And when she looked, the output of the vertical sensing system was right up against this bound. Moreover, triplets that come from that sensing system also sit near the bound, though they slide a little bit further down. It's just 3 out of 10. But the triplets are the putative things that project downstream to the motor center. Another thing Siwei could do, because she's in silico, was rip the circuit apart and find out what gives it this behavior. So one key thing she found was that these axonal gap junctions in the vertical sensing system are what make the difference. So when she takes those out, you fall away from the bound. Now, or you fall so far down the bound that you don't have enough information to resolve your heading direction. And that is what she found. She also found this is the how much prediction is there and what is it encoding. She found that it's encoding your heading direction at fine degree of visual angle. So these are different projections onto principal components of a sort using a variational information bottleneck technique that help the fly resolve heading. So heading in sort of this direction versus that direction versus that direction. These all splay out in very discriminal different clusters in this space. So the output of the fly visual system is able to do some linear decoding of these heading directions. And that's what we think helps the fly know not only the escape path it needs to run, but also where that will play out relative to its body position in space at the time of the escape. So it needs to not only know where the threat was and like play the motor tape forward that lets it move away, it needs to know where its body was relative to the gravitational field and how its body was oriented. And putting all that together means that it needs to be able to tell at a very fine scale where it's heading. And that gap junction, axonal gap junction, lets the fly do that. Okay. So those are several different vignettes from across some vertebrates and even this invertebrate that really say that what you need to do, the behaviors that are mission critical for the organism, shape the early visual sensing system, potentially for this kind of ubiquitous computation, which we call prediction. I want to take 2 minutes to just entice you into conversation about some future directions and where we might elaborate this further. So we all know that the retina goes many, many places in the brain. So retinal neurons, we've talked about them going to colliculus today. Of course, we know they go to thalamus, they go to the lateral geniculate nucleus. They spread out all over the brain. And all of these different projections might be carrying different information about the visual scene. There are many computations that follow on from what gets computed in the retina. We also know there are all those functional classes that we talked about at the beginning. So what's optimal and what you need to do with that visual information could be very different for different populations of cells. And natural scenes are full of complex features that could be relevant to the organism. So this is one of the movies from our natural motion database. And there's a lot going on here. There are bees that are tending to larvae. There are bees that are keeping the whole hive cool. There are bees that are carrying out a dead body. There's somebody who comes in and does a waggle dance at some point. There's lots and lots and lots going on here. You could even discriminate the identity of these bees and track them if you wanted to, throw all the awesome power of machine learning at this. So there are lots of things that maybe the bee brain could care about in this, or that us as observers, our brain could care about. If we just take the motion aspects and we project this onto an optic flow field where color is direction and saturation is speed, then we get a movie like this that shows us, just the motion component. And now we've lost the hive. We maybe don't even know what species of bee these are. And we could play that against a white noise background and ask, different parts of the visual brain, do you care just about this motion, even if there are no bees in any individual frame? And so there are many, many things that the eye could be peeling apart here. And we don't think that motion and prediction are the only things. They were just our best guess at something that is important and ubiquitous. But I think that as a community, we can tap into more and more interesting computation and find more examples of optimality if we shape that notion of optimality by the output behavioral goals of the system. So we have to dig into the biology more to do that, but I think it's an exciting way to go, especially given that we have such a massive amount of recording going on on behavior and in natural scene stats. So I hope I've told you these three things. I hope you're convinced. But either way, let's have a conversation more about this. I'll just wrap by thanking our funding sources, by thanking my incredible group of people who do all this work, and definitely keep an eye on these names, because they're going to be running off and doing their own independent things, and it's going to be amazing. And I'll take your questions.
Wow, that was fantastic, Stephanie. Thank you so much. For you that might be watching this on demand because of bank holidays or whatever, and you feel like you are missing on a great opportunity to ask your questions, too bad. I guess maybe Stephanie is all right with you sending an email with them.
Sure, of course.
And like for the ones that you might be there and feel very shy to ask your questions, I'm right now posting the Zoom room link for the conversation that will continue offline in a couple of minutes from now. So the first question that I have personally, Stephanie, in terms of, because you kind of hinted at it, like with the different notions of optimality, you also mentioned at some point in your talk that, for different stimulus parameters, you observe like that we are quite far away from the optimal border, right? So the question that I have is like, maybe starting also this discussion, like, do you think that in order to talk really about optimality, we need to kind of regard the stimulus space in a more broader sense, and whether like optimality for the retina doesn't necessarily mean optimality for the brain or for the behavioral output and so on?
Oh, absolutely. I think that taking the complexity of natural scenes and natural inputs seriously is what gives you ideas about where things have evolved to. So there's a massive space of different kinds of motion that here, let me stop sharing for a second so I can do big hand motion. So there's a massive space of different kinds of motion that the brain could have evolved to do good prediction on. And if we make decent guesses about where the brain kind of sits, so that damped harmonic oscillator motion isn't completely crazy and isn't completely arbitrary. You can think of it like a leaf that's tethered on a stem that's getting kicked around in the wind. Right. And it's got some mass. And so we can think about it as like maybe a background motion. Maybe you want to be able to predict that so you can subtract it away. But of course, we want to find the edges where that doesn't work anymore. We want to find the places where the motion is, different from that. And so what you'd like to be able to do is to sort of enumerate all of the different kinds of optimization you get and what you don't get. We're not saying that would be the end of the day, but at least that would lead you down a path towards elaborating that a bit more. We kind of want to sit in between drifting gratings and natural movies. And if we sit over here with natural movies, we can make some progress in trying to extract out essential motion elements, but playing it all at once is really complicated. So that's what we've been trying to do to shape to shape that path and exploration. So we put some flags down where we think there's good further exploration to be done.
Right. And before I move on to a question that Tom posted, so going back now to the cell properties, right? You mentioned that for salamander, you have off-dominated, for mice, you have on or on-off, and so on. So I was wondering, okay, so this looks like in terms of polarity, it doesn't really matter what optimal encoding is for a certain stimulus, right? But I was wondering, what about other properties like transients versus sustained, or, you know, firing with a certain latency and so on? Would you expect these differences?
It's a fantastic question. We have anecdotal evidence that of some of those things, but we need a much bigger survey to suss that out. I think it's exactly the right question. We found so little bits of anecdotes, right?
Mm-hmm.
So we found that when you have cells that have different functional classes, they actually form really good predictive clusters. We also find good prediction within a functional class. So it does seem like you can kind of do it either way, but asking yourself then the biology question of where do these things project? Is it reasonable that they'd be synapsing onto the same neuron that can collect this information together and process it in this way? Those are the right questions. There are things that are even simpler that we found, that if cells are the same functional class, but the receptive fields are separated a little bit, those can be good predictors for obvious reasons, that
Yes.
if you have some integrator that can see across space, that's wonderful. Even more interesting is when you have an on cell and an off cell that are on top of each other. Then when they fire together, you've got some interesting feature. You know that if one fires and the other didn't, then maybe the bar is leaving that space or something like that.
Yeah, of course.
So, you know, when you consider arcs in time, I think you can get all kinds of stuff. But the short answer is that you have to go back and think about where things are projecting and what's going on downstream to really tease that apart.
Right.
And I think there are some subclasses that won't be predictive, and that'll be just fine.
Yeah, being there for different reasons. And if no more questions appear, I think this will be the last question, given that people are already gathering here, so we can continue offline. So the last question that is currently in the chat is from Tom. Can you elaborate a little on how prediction tallies with experience or learning or statistical regularities in the world? For example, you get better at predicting stuff the more you do something or you experience something.
Yeah.
And this is a question both on individual level and over the course of evolution. So what knobs does this act on?
Oh, that is fantastic. I mean, that's a whole research program right there, Tom. That's fantastic. So, you know, the retina has evolved all kinds of adaptation mechanisms. And I think that's the key to knowing how that evolutionary knob sculpted the suite of things you can flexibly change your response to. So how much can you change your prediction paradigm as the motion in the world changes? And I think for the retina, that's not going to be an individual experience thing. That's going to be an evolutionary experience thing. And what would be really nice is to write down a theory about how the suite of adaptation mechanisms supports this class of motion prediction and what the edges of that are, so where it fails. And then downstream, I mean, I think that's fantastic, because prediction is one word, but it embodies many different kinds of computations on many different length scales and time scales. So downstream of the retina, you could imagine learning. We've had fun showing that you can learn to be optimally predictive with just short time scale spike timing dependent plasticity. That was Audrey Cederberg's work when she was a postdoc. She's now faculty at University of Minnesota and moving to Georgia Tech. So congrats, Audrey. And I think that's exactly right. Like how would you learn and change with experience? That's got to happen downstream.
Right. Thank you very much for the fantastic talk and for addressing the questions, Stephanie. I'm going ahead and posting the Zoom room link again in case people cannot retrieve it from the first time I posted it. And I think at this point I will be terminating the broadcast so we can continue in the traditional, more informal fashion over here. Thank you.
Thanks, George, and thanks, Tom.
So we are officially